So let's go ahead and talk a little bit about amino acids. You've learned a long time ago that there are 20 standard amino acids, and the differences in them are the R groups, or the side chains. And so if we look at this structure here, we see that this is a general structure of amino acid, be able to draw this and know what each of these represents. And so every amino acid has an amino group and a carboxyl group, and that amino group and carboxyl group are formed through a condensation reaction, right, with water being released to form a peptide bond. And so peptides, which have multiple amino acids, in this case 4, you can see that they were joined by peptide bonds. Now this peptide bond is rigid and it is planar. So if we look at this representation of this peptide, we see that we can shorthand this for alanine, tyrosine, aspartic acid, and glycine. So I expect you, through the course of the semester, to know the abbreviations for all 20 amino acids. And we'll work on them in groups as we need to. But what you should start understanding is that when you see these on a test, on a quiz, on homework, you need to be able to understand which amino acid it is. So the first kind of topic we're going to tackle now, in terms of getting right into the real material here, okay, the other was real, are pKa's. And no surprise, we've been working on pH and pKa's now, right, since the beginning of the class. And so it matters. And so again, remember why understanding acid-base chemistry, pH, pKa matters for a lot of things. One, when you're making buffers in lab, you use that so that your protein is stable. In the lysozyme lab, you're starting out with a tris buffer at a pH that is tuned to be below the pi of lysozyme, so that lysozyme is protonated and charged, so that it's charged and has a charge so that you can separate it by cation exchange chromatography. So there are a lot of reasons, and the one for the side chains that I want to talk about right now is enzyme catalysis. Okay, and so as part of one of your labs, you're relating how the local environment and enzyme can affect the amino acid side chains, such as histidine and aspartic acid in this particular example, to then be involved in the actual reaction mechanism. Okay, so I just want to talk about the fundamentals here. So here what we've got are the terminal carboxyl group and the terminal amino group, and that is these right here. That's what we're talking about right there. So if we look at these pKa values, we see that they do vary, and we understand why, even though they're all carboxyl groups, why they can vary. And we talked about that. One example we did on the board together was the difference between the pKa values of oxalate and succinate. So apply that here, right? Same with the amino group pKa. And so what I want to hone in right now are the ionizable side chains, and let's relate it to the pH of physiological pH. And there are different compartments in different cells that do have different pHs, of course, but if we just go with pH 7.4 for physiological, what I want to ask you right now is which of these amino acids, right, are going to be charged at physiological pH. So if we look at aspartic acid and glutamic acid, right, if we look at that, pH of 7.4 is above this, right? And so we know that these are going to be, a lot of them will be unprotonated. So they will be in this negative form here, right? So they'll be negatively charged. If we look at lysine and arginine, they're way above physiological pH, right? And so in this particular example, since they are existing at pH 7.4 in our hypothetical example, we know that they are going to be positively charged. Okay, and you could use the Henderson-Hasselbalch equation for that, but these are pretty easy to understand. Now tyrosine, you may say, Dr. Willett, this is also going to be protonated, and it is, but if we look at it, we're not looking at something that's going to be charged. Most of it, tyrosine, is going to be protonated, so it's going to have the hydroxyl group. If we look at histidine and serine, they're both 1 to a little bit more away from pH 7.4. So histidine with the pKa of below 7.4 means that it is going to be mainly in the charged form. And if you use the Henderson-Hasselbalch equation, and I want you to, pH equals pKa plus the log of the molar concentrations of the weak base divided by the concentration molar of the weak acid. And remember, when you just do the mathematical manipulations for this, when you plug in the physiological pH of 7.4, when you plug in the pKa and do the mathematics, you then get the number for the ratio of the weak base over the weak acid, and that can tell you the percentage of each. And so when you do that for histidine, what you see essentially is that there's about 8% that are charged. And so if we look over at histidine, about 8% are charged, which means 92% are not charged. And if we look at cysteine at 8.37, what we see is that about 10% are deprotonated. And again, go ahead and look and use the Henderson-Hasselbalch equation to get that. So you should be able to determine that, right, on your own at this point. So please do that. And so the reason that matters, of course, is because now that we know that lysine and arginine are charged, they've got a positive charge at physiological pH, and that aspartic acid and glutamic acid are negative, we're starting to understand some fundamental principles about how enzymes are going to react when we get to that stage. Okay. Same thing with histidine and cysteine, since they're closer, right? They're able to fluctuate back and forth between the protonated and the non-protonated much more readily. And so we'll get into that a little bit more. So if we just look at, without looking at the amino acids, side chains, if we just look at this particular example right here of an amino acid, and we just look at the protonation and deprotonation, what we'll see is that the deprotonation of this, for pKa1, is the deprotonation of the carboxyl. And if you go forward in the titration curve, then we're looking at the deprotonation of the amino group. And so the key thing to know about here is that essentially what we're looking at is at neutral pH, or around pH 7.4, what we have are charged, both of these charges, and the net charge is 0, right? And so the net charge is 0 it's called a Zwitter ion, meaning 2 ions, 2 different charges. And so what we see here is that in this range, including the physiological, we have a lot of this right here, this Zwitter ion. And so this is an important concept for you to understand. And so basically, what you're also going to determine is we're going to relate this to the isoelectric point. Okay, but understand that. So we've been doing titration curves. So this right here, you should be able to know this like the back of your hand, right? And so if we then look at, so say you are titrating alanine, okay, like you're doing in lab, you're titrating 2 amino acids, and we see that here we've got the first equivalence point, and we've got the second equivalence point. And so we know that this is the first pKa around this region where we have the largest buffering region, and then this is the second. Well, in the case of alanine, its side chain is not ionizable, so the only ionizable groups that we're looking at are the alpha amino and the alpha carboxyl group. And so, of course, this should be the alpha carboxyl group. And if we look at that, we can look over here to this image where it starts out with 1 charge. As you titrate it with hydroxide, and now you get here, you're starting to build more of this. And when you're at the isoelectric point, right, you're essentially at where you've got all of this, right? So here it's 50-50, here it's essentially 100%, right, of that PI. And so at that first equivalence point, in the case of these amino acids which do not have ionizable side chains, that PI is between the first 2 pKas. Well, it's between the 2 pKas, pKa1 and pKa2. And so to get that pH, that pH value, it's just the average of these two. So 9.69 plus 2.34 divided by 2 equals the PI of, in this case, alanine. So PI is a really simple concept. It's the pH where whatever ionizable groups are on there, they all balance out so that you've got essentially well, no charge, no net charge, although you do have 1 positive and 1 negative charge. And so if we look at that for groups with ionizable side chains, right? So I've got this listed here. So let's do a search for histidine titration ChemLibra. Right? So this is a great website. I trust it. And if we go to this 26.2, what we see is this group has shown us, although I put in histidine, let's go ahead and look at arginine. So this is a titration curve of arginine. And so now there's 3 pKa's. So how do you determine the PI of when you've got 3? You don't just take them all and divide them by 3. That's not how this works. Think about the molecules that are happening, right? It goes from as many protons to as few protons as you move, well, you're looking at me, as you move from low pH to high pH as you're titrating it. So when we look at this then, what we see is that when we start out the titration curve at low, pH less than 2, we've got 2 positive charges on it. So it's not at the isoelectric point because it's plus 2. If we then go up to 0.8, then we've got 2 pluses and 1 negative, so it still has a net charge of plus 1. At around pH 10.8, aha, look at that, 1 positive and 1 negative charge. And then here we've got negative charge. So at this particular example, right, here, what we then do is, it says 10.8, but you wouldn't know that yet. You're trying to figure that out. But when you draw this, because you say, okay, it starts out fully protonated, right? As it moves over here, it moves to 100% this. As it moves over to here, it moves to 100% this and then 100% that. And so when you realize then that this right here is going from this pKa to this pKa for here, then you say, okay, it is the average between 12.5 and 9, and that's the approximate value you get. So in other words, you average the 2 pKa values that are on the opposite sides of the structure that at 100%, right, that equivalence point has a net charge of 0. And so what you do is you draw out the structures and you look to that point where like, the number of positive charges balances the number of negative charges. Okay, so we know that some amino acids have charges, right? And so, so those would be acidic or basic. We know that some are polar and some are hydrophobic. And so these are what they vary in, and we'll sort them into these 4 groups. So be able to do this. If you see an amino acid, if I give you a structure, be able to tell me which group it falls in and why by describing it. Okay, so now we're just going to go through some of these amino acids kind of quick. We're not going to get heavy into them. As we progress through the course, we'll understand these more in context, okay? But you definitely need to know the aliphatics are these kind of open chains. They're non-aromatic. And so if we look at this, glycine, alanine, proline, valine, and leucine. Okay, so we have different structural representations, and what we see then is that, for example, all of these amino acids except glycine are chiral, and we will talk about chirality, but basically what that means is that the carbon atom must have 4 different substituents attached to it. And so in this case, the fact that this carbon has 2 means that it is achiral. Okay? Whereas if you look at this, it's got a methyl group, a carboxylate group, an amino group, and a hydrogen, so that is chiral. And every one of these that you look at, you will see that that alpha carbon that you drew, or you will draw here, right? Here we go. 1 substituent, 2 substituents, 3 substituents, 4 substituents. So as long as that's not hydrogen, then it would be chiral. And so when we say alpha carbon, that's what we're talking about here. And so these are hydrophobic, and so the question is, why are they buried in a protein's interior, right? Think about the discussions we've had of hydrophobicity and the energetics based on entropy, for example, when we talked about looking at alkanes and the ability for it to move from a water solution into an organic solvent when you shake it up, right? And so all of that comes into play here, and that will be put in the context of inside a protein. And so when we look at this then, all of these, there's no polar groups on this, are there, right? This is just a bunch of carbons and hydrogens. Now this is an odd one we'll talk about later, but let's not worry about it for the moment, proline. And so this is just showing some of the sizes that the larger you get for these nonpolar, then you've got these large, big kind of hydrophobic groups. And this one, and this one, and this one all differ in that 2 of these are aliphatic and 1 is aromatic. So which of these is actually aromatic? Yeah, it's phenylalanine, that aromatic group right there. And so back to the aliphatics, what we're seeing here is that, again, CH is carbons and hydrogens. Here, methionine, we actually now have an electronegative ion. We still consider it aliphatic, and this is because it's a thioether, so it is really not very reactive, so it kind of behaves as a CH2 group. It's a simplistic version, but we'll stick to that for now, okay? So the aromatics, we already saw phenylalanine, and then tryptophan gets even bigger. Okay, and so when we're looking at this, and really let's see, did I talk about these? No. When we look at phenylalanine, this was named because it's got a phenyl ring, which is this, and it's like on an alanine. So alanine would be just CH3 right here, but so phenylalanine is replace one of those hydrogens with a phenyl group, and you've got phenylalanine. You can see that through here. And then tryptophan, this group right here is an indole group. That's the name of it. And then methylene group. And so, so that's important to know. Okay? And we'll talk later about why that's less hydrophobic. So in terms of polar groups, we've got hydroxyl groups and we've got sulfhydryl groups. And so if we look at serine, we have, again, here's the alpha carbon, here's the amino acid, and then it's got CH2OH. And that's this right here, and that is this right here, CH2OH. So it's polar, right? Polar hydroxyl group. Threonine is kind of the same as serine, but instead of just CH2, it's now got a methyl group. Okay, so and if we look at this, this is starred there because now if you look at this, this is also an asymmetric carbon, right? It's got 4 different substituents on it. So that's unusual for the side chains. And then tyrosine here, it's like alanine plus phenylalanine plus a hydroxyl group. So if you were to remove this hydroxyl group and put a hydrogen there, that would be phenylalanine. So the only difference between tyrosine and phenylalanine is that hydroxyl group. But that hydroxyl group is really important. Okay, and so this cysteine group is essentially a serine, but instead of oxygen, it is a sulfide. It is a sulfur group, and so we call that a sulfhydryl group or a thiol. Okay, so this is more reactive than a serine group, and the reason that it is more reactive is that, well, let's see, maybe we'll talk about this in terms of pulling off the, well, let's see, if this is, let me put it this way, it's more reactive, it forms disulfide bond, which we'll talk about, and in terms of the pKa's, that's what I should do. Let's go back and talk really quick about these pKa's. Here we go. So if we look at cysteine pKa at 8.37, well, where is serine? We're comparing it to serine. Now, serine's not even on here because it's way down below. Okay, it's higher than this. So serine, by and large part, it's not losing that hydrogen group, but cysteine, as we talked about, can lose it. Now, the reason cysteine is more acidic than serine is, and it's actually, I think, shown here, maybe not. This is, cysteine is actually bigger. Excuse me, the sulfur group is bigger than the oxygen. So in terms of releasing a proton, the fact that it is bigger, that negative charge can be separated out or scattered more on a larger area, larger volume, okay? And so it is more stable. And remember, if something is more stable in one condition versus the other, that's a decrease in energy. The Gibbs free energy will be lower, and so that will be the favored energetic, well, not quotes, that will be the more energetically favored structure. And so cysteine is more acidic. The pKa is much lower than serine because it is bigger and allows that negative charge to be more stable on that. And so in terms of acidity, if you go down the periodic table, oxygen, sulfur, and you keep going down, that's how you look at that particular example. And so more polars, what we have here is essentially a carbonyl group and an amino group, and that's called a carboxamide or just an amide group, okay? And so, so that right there, very polar, right? So NH2, that is a polar group, and the carbonyl group is a polar, so combined, they're certainly polar. And so what we see is we've got a more basic group and a more, not more acidic group, but a more electronegative group. So asparagine and glutamine, very similar. It's just that glutamine has this extra CH2 here compared. So this is CH2, This is CH2 and then the carboxamide, and this is CH2CH2 carboxamide. And so we just talked negative charge because of that physiological pH, 7.4, much higher than the pKa of these carboxylate groups, and so they are negatively charged. And these are just some ways you can think about calling them, right? So positively charged, arginine, lysine, and as we talked about, histidine, right? And so arginine and lysine are essentially almost 100% of these at physiological pH are charged, as opposed to histidine, which we talked about 8%. But still, and it's not drawn charged here, but we could draw that. We could throw a hydrogen on there and protonate it. Okay, and so this group is a guanidinium group right here. This is a primary amino group, and this is an imidazole ring. So you need to know these terms. These are some basic fundamental terms that you need to understand. So this is lysine, and so you can see it's got this kind of straight chain, and we've got this basic group at the end. So cysteine, one of its very special properties, and this is really important for a lot of reactivity as well as some stability in proteins, is that it forms disulfide bonds. So if we look at this then, if this is one polypeptide, this is another polypeptide, and they come into close enough contact that they can essentially, let me ask you this, from going from A to B, is this an oxidation reaction or a reduction reaction? Is it oxidation or reduction? Well, yeah, it's got this oxygen group here, so it's an oxidation reduction. And you can also see that these hydrogens are going away, right? So if we go the other way, even if you didn't see the oxygen and the water here, you would see that these gained hydrogen, so that's a reduction going in the reverse reaction. So disulfide bonds are important. We will talk more about them. Cysteine is the only amino acid that it can do that, and it does it with, well, not itself, but another cysteine. So histidine, an example of kind of applying that physiological pKa, right, where 8% are protonate. So if we look at that then, this allows histidine to gain a proton and then to release it. Gain and release, gain and release. And we'll talk more about that, but think about it. It's a shuttle, it's a handle, it's something that can gain a proton or during catalysis gain a proton, and that's part of one of your labs that you're working on, okay, is the ability to be protonated at certain pHs or the ability to stay unprotonated at certain pHs. So this checkpoint, I like these checkpoints in your book, so I've assigned checkpoints. You see that on Blackboard. So do this, right? This is active learning. Complete all of the checkpoints that are assigned. Make sure you can do this. Even some questions you'll see something close to on a test, but all of your questions from the test aren't just going to come from the book, of course, right? So, but these will help you succeed in questions that I write myself. Okay, so same with here with the key concepts. Definitely understand this very clearly. So this optical activity, let's go ahead and make this another lecture.